This is the story of CRISPR-Cas9 intellectual property rights. To understand where we are today, we need to go back to track 3 stories. 2 of those stories have seen wide exposure to the world, the story of Emmanuelle Charpentier and Jennifer Doudna. Let us start with a brief background story of Emmanuelle and Jennifer and the discovery of CRISPR-Cas9. Emmanuelle Charpentier grew up in Paris. She liked pure science and mathematics, but was also interested in human sciences like psychology, sociology, and philosophy. She thought that her father teaching her the Latin names of many plants in her childhood possibly influenced her to pursue science, and we sure are glad that she did. Charpentier attended the Pierre and Marie Curie University to complete her undergraduate and postgraduate studies and received a degree in biochemistry in 1991. Later on, she went on to do a PhD in microbiology in 1995. Then Charpentier went on to do her graduate research at the Pasteur Institute. She was excited to be a scientist. At the Pasteur Institute, Charpentier investigated the genetic and molecular mechanisms behind antibiotic resistance. Soon her focus turned to the study of how bacterial pathogens infect and interact with their hosts and environment, focusing on the molecular mechanisms during infection with the aim to find new pathways that could be further harnessed for the benefits of medicine. Charpentier also developed tools to facilitate the genetics in bacterial pathogens and studied proteins involved in cell-to-cell adhesion and signal transduction by developing transgenic mouse models. Later, she moved to the United States in 1996, where she worked in a research associate position at 3 New York City institutions. The first one was the Rockefeller University, then the New York University's Langone Medical Center, and the Skirball Institute of Biomolecular Medicine, as well as St. Jude's Children's Research Hospital in Memphis, Tennessee. 6 years later, Emmanuelle returned to Europe with all the gathered experience to establish her own research group at the Max F. Perutz laboratories of the University of Vienna in Austria in the field of microbiology. Soon she became associate professor and then guest professor at the Laboratory for Molecular Infection Medicine, Sweden, also called as MIMS, at Umeå University in Sweden, where she habilitated in the field of medical microbiology. Since 2013, Emmanuelle Charpentier is the head of regulation in infection biology department at Helmholtz Center for Infection Research and professor at the Medical School of Hanover in Germany. It was at Umeå that she developed the CRISPR-Cas9 project. One RNA, tracer RNA, retained the attention of her laboratory. This RNA was located in the vicinity of odd DNA sequences known as CRISPR, or clustered, regularly interspaced, short palindromic repeats, CRISPR in short. When a virus attacks the bacteria, a snapshot of the invader's genetic identity is stored on the CRISPR sequences and then used to repel the virus should it attack again. CRISPR-Cas systems have evolved extensively in bacteria. No one knew exactly how the process for the so-called type 2 system, which we know as CRISPR-Cas9, works. In 2011, Charpentier published a paper in Nature that identified the critical role that transacting CRISPR RNA, that is tracer RNA, performs in development of CRISPR-mediated virus defense. Jennifer Doudna was born in Washington. Her early childhood was in Hawaii, where both her parents were teaching. A copy of James Watson's The Double Helix, which her father gave her when she was in the 6th grade, sparked her interest in biochemistry. Doudna did her undergraduate at Pomona College in California, which had a strong biochemistry program. Later on, she went to Harvard for graduate school, where she worked with the future Nobel laureate Jacques Shostak, who was researching RNA, one of the 2 types of nucleic acids found in all cells. Doudna helped engineer a self-replicating catalytic RNA that reinforced her hypothesis that RNA, not DNA, was the source of early life, for it demonstrated that RNA not only stores genetic information, but also initiates the chemical reactions that help duplicate the information. Subsequently, for her postdoctoral studies, she went on to the University of Colorado at Boulder to work with biochemist Tom Schech, who had just received a Nobel Prize for his discovery of the catalytic properties of RNA, where she commenced working on the three-dimensional atomic structure of those molecules. This resulted in Doudna and Schech publishing a landmark paper in 1996 that described for the first time what a structured ribosome looked like. In 2002, Doudna moved to the University of California, Berkeley, where she continued to study the molecular structures and functions of RNA molecules. Later in 2005, Doudna came across a rare type of bacteria that had in its genome an unusual repeating sequence called clustered, regularly interspaced, short palindromic repeats, or CRISPR in short. The sequences enabled the bacterium to remember the genetic identity of a previous viral attack and then use that memory to mount a successful defense against the virus. Doudna was intrigued and decided to figure out how the process occurred, publishing a series of papers over the next few years about the molecular functions of CRISPR systems. The story of Charpentier and Doudna intersects in 2011 at a conference in Puerto Rico. That's where they met for the first time. Charpentier wanted to have the structure of the Cas9 complex to investigate the possibilities to reduce the system to its minimal and identify the position of all active domains in the structure of the complex. She felt that her work was complementary to that of Jennifer, and collaborating would make great sense, and that made working together even at such long distances very enjoyable. They decided to pool their expertise, biochemistry, structural biology, and microbiology to determine how CRISPR provides bacteria with viral immunity. And then their work bore fruit, and on 10th of December 2020, the world saw the announcement of a Nobel Prize to Jennifer and Emmanuelle for the discovery of CRISPR-Cas9.
I feel extremely honored to receive this highest recognition from Sweden. I think surely of my former team in Vienna and at Umeå University, who contributed to this great discovery. I also think of all my colleagues of the CRISPR field and all the scientists who have really done a fantastic job to bring this technology further. That allows now to have a genome engineering technology, a very versatile one, that is used by multiple scientists all over the world to modify genomes of cells and organisms. So it's a great discovery, and I'm very happy to be one of the pioneers. I find it for sure extremely nice that the award is going for two women. I think this is the first case ever seen for the Nobel Prize. I think it's quite also reflective of what you see nowadays. One has always to keep in mind that in general, Nobel Prizes are awarded 20, 30 years after the discovery. And this discovery is only 8 years ago, and it has bloomed extremely. So everyone is using now the CRISPR-Cas9 technology, and I think it's just reflective of what is happening in our days, meaning that scientific work involves collaborations, and we collaborate with scientists we think we should collaborate with, whether they are female scientists or male scientists. Here it's just happened that it's two female scientists, and it's quite reflective of what is happening in our days. I hope it will remain as such, but it's reflective of the fact that science becomes more modern and involves more female leaders.
With that came instant worldwide recognition and a major boost for CRISPR-Cas9. This is the most well-known story of CRISPR-Cas9. However, this is not yet the complete story. With many researchers working worldwide to crack the mysteries of science, human history is full of cases in which several researchers have found the same or similar inventions almost simultaneously. This seems to have been the case with CRISPR-Cas9. To understand the IP battle, we need to understand the timeline of the CRISPR-Cas9 patent application. In May 2012, Doudna's team, including Charpentier at the University of California, Berkeley, filed its first patent application for CRISPR-Cas9, the first of many to follow at the US Patent Office. A few months later, in December 2012, a separate research group at the Broad Institute, affiliated with MIT and Harvard University, also filed its first patent application for CRISPR-Cas9. We need to understand that it is normal for institutions to file a multitude of patent applications which kind of remain secret for approximately 18 months. Only when the curtain got lifted did the institution realize that they have filed competing patent applications for CRISPR-Cas9. The same drama played out at the same time in Europe, but in reverse order. In March 2013, UC Berkeley filed a patent application with the European Patent Office, claiming priority from its US patent application filed in May 2012. Shortly thereafter, in December 2013, the Broad Institute filed a competing application with EPO, claiming priority from its own applications filed in United States in December 2012. The universities also remained in limbo in Europe as they awaited the publication of their respective applications. As of 2016, in the United States, UC Berkeley and Broad Institute launched priority proceedings to be granted the exclusive rights to CRISPR-Cas9 technology in the form of patents for invention. Basically, UC Berkeley claimed broad general applications, whereas the Broad Institute claimed a more narrow eukaryotic cell application pertaining to plant and animal. At the US Patent Office, they were considered to be distinct, so UC Berkeley filed an appeal but lost to Broad Institute in USA. However, Patent Trial and Appeal Board, PTAB, announced that a third hearing would be required to determine the priority of the patent filings. All jurisdictions in the world where patent application claim priority from either UC Berkeley's or Broad Institute's US patent applications are therefore awaiting the outcome of the proceedings to determine which competing patent applications may be granted. However, on the European patent front, UC Berkeley won due to an error in filing by the Broad Institute. The outcomes of appeal in the US will influence the decisions of stakeholders in Canada and other countries, and the decisions of examiners at the Canadian Intellectual Property Office, CIPO. In the meantime, there are more than 1,200 patent applications pending before the CIPO regarding the CRISPR-Cas9. In order to commercialize new CRISPR-Cas9 technologies and applications, interested parties will need to obtain commercial licenses to the basic CRISPR-Cas9 patents. However, this task is complicated by the fact that licenses must be obtained from different sources. The owners of the core patent applications have granted their rights exclusively to marketing companies with the mandate to grant exclusive or non-exclusive licenses to private companies willing to invest in developing applications using CRISPR-Cas9. CRISPR Therapeutics obtained its exclusive rights from Emmanuelle Charpentier, Intellia Therapeutics from UC Berkeley, and the University of Vienna, whereas Editas Medicine from the Broad Institute. For all other areas, the companies holding the relevant rights are ERS Genomics, Caribou Biosciences, and the Broad Institute. ERS Genomics obtained its exclusive rights from Emmanuelle Charpentier, Caribou Biosciences from UC Berkeley and the University of Vienna, while the Broad Institute licenses CRISPR IP non-exclusively for commercial research or to companies wishing to sell tools and reagents for genome editing. We will therefore have to wait for a commercial agreement between the holders of potential IP rights on CRISPR-Cas9 or for the outcome of the US legal proceedings In the meantime, scientific research using this revolutionary technique continues to make great strides. Apart from just CRISPR-Cas9, we have even more proteins that are in play right now. The first one is Cas1, then we have Cas2, and then we have Cas3, which was discovered in 2002, and then we have Cas4, we have Cas6, we have Cas7 to 11, which is called as Cas-mini, which is again a important one. This was designated as Cas-mini by Stanford University and has just 529 amino acids. That makes it a much smaller molecule than CRISPR-Cas9. And in their own words, what they are saying is that if Cas9 is a scissor, Cas-mini is a Swiss Army knife, smaller but with many functions. So this is going to be much more superior to Cas9 for many applications. And as such, the delay in granting patent rights is just piling on more and more risk for various patent users. And the sooner the patent issue is resolved, the better. And if we were to look at what else is in play, we have Cas12, 13, and 14, which is currently being used by Mammoth Biosciences for diagnostics. But in my mind as an investor, I'm thinking that CRISPR is the original concept, and CRISPR and Cas9 combination encapsulated the concept of the tool, which could be used to edit DNA. So anything else that comes after that is building on top of CRISPR-Cas9. So even though a company may not use Cas9, they are still working on CRISPR. And as such, I think that with more and more Cas proteins being discovered and put into use, we are getting into a situation as investors to be very alert on the judgments that are coming about the patent ownership, because to me, it is still very murky. I did a research on this for almost a week to 10 days. To summarize everything, I would state that the core IP is either with UC Berkeley or with Brock Institute. These are the two contenders. The first one to file was UC Berkeley. However, they filed for a much broader scope. They were very general in their description, whereas Brock Institute were very specific about what area their discovery is going to cover. And because of that, initially, the patent office thought they were two separate applications. So that's where the initial problem has started in terms of determining ownership of the IP. So we'll have to wait and see what happens. Any company that derives their IP agreements directly, IP rights directly from either Brock Institute or UC Berkeley is likely to have much less of a risk as compared to any other company which derives through third parties. So that's my conclusion overall. This video is likely to get dated very quickly because there is continuous development, and there may be the third hearing and judgments which will come through. So stay tuned on this area if you are a CRISPR investor, and continue keeping track of news items. As soon as I find out anything, I'll definitely be the first probably to bring it to you in this channel in one of our CRISPR updates on our daily update videos. So thank you very much for listening to this story and for watching this video. I'd like to see you again in the next session. Till then, bye for now. Please don't forget to put a like, and if you're new to this channel and here for the first time, please subscribe to this channel, and you can expect to see a lot more on CRISPR.